Hi, good morning. This is Kavita Kundra, and welcome to the next chapter of Mscdfsm Biochemistry. The chapter name is Vitamins. Vitamins are the organic compound required in very small amount for growth and maintenance of body. But if there is any deficiency occurs, they result in serious diseases. In 1880, Sir Frederick Crowell and Hopkins reported necessity of factors. In 1912, Schwank observed diseases like beriberi, scurvy, pellagra can be prevented by food, and he proposed the name of these substances as vitaminae. As all compounds do not possess nitrogen, so the name given was vitamin. Vitamins are synthesized by plants, and some vitamins are synthesized by gut microorganisms of the animal. Example, a rat synthesizes vitamin C, but guinea pig cannot. Human can synthesize vitamin D and niacin. Types of vitamins: water soluble vitamins, fat soluble vitamins. So there are two types of vitamins. One is fat soluble, another one is water soluble. Fat soluble vitamins are A, D, E, K, and all others are water soluble like vitamin B1, vitamin B2, vitamin B3, niacin, B5, B6, B12, vitamin C, vitamin H, and vitamin M. So what are water soluble vitamins? Water soluble vitamins, we first learn about thymine. What is thymine? It was isolated in 1926 from the rice polishing by Jensen and Donath. In 1931, its empirical formula was established by Windows, and in 1936 the chemical formula was elucidated by William and co-workers. Thymine occurs in four forms. First, thiamine. Second is TMP, thiamine, M for mono, P for phosphate, thiamine monophosphate. Then TPP, thiamine diphosphate, and thiamine triphosphate is TTP. What are the properties of thiamine? Thiamine hydrochloride is white needle-shaped crystalline substance. It smells like yeast. Thiamine has sulfurous odor and bitter in taste. As thiamine is a water soluble, so because of this property, it is easily lost due to washing, soaking, and other cooking processes. Moisture increases the destruction, so it is less stable in fresh foods as compared to dry foods, and it is stable in acidic medium at room temperature. This compound is converted to inactive derivative thiochrome by controlled oxidation. Thiochrome has strong fluorescence. Thymine, when dissolved in sodium bisulfate at the pH 5.0, it gives pyrimidine and thiazole. And this property is used for chemical estimation of thymine. Second is riboflavin. Riboflavin means vitamin B2. In 1933, it was isolated by Kuhn from milk. So first it was named as lactoflavin because it was from milk. Riboflavin has isoalexoxyzine nucleus, that is piperidine ring with benzene ring fused to it. The side chain is C5 polyhydroxy group, that is a derivative of ribitol. Chemically it is known as 6,7 dimethyl 9D ribityl isoalexoxyzine. Riboflavin has two major coenzyme derivative, FMN and FAD. So what are the full forms of FMN and FAD? These are very important from examination point of view. FMN is flavin mononucleotide. FAD, flavin adenine dinucleotide. What are the properties? It has needle-shaped orange cluster. It was earlier was white. It was orange. It is sparingly soluble in water. Aqueous solution of vitamin B2 imparts yellow-green fluorescence, and it is stable in boiling in acidic medium but destroyed by heat in alkaline medium. It is sensitive to light. That is the reason we should not leave milk in sunlight, as milk has riboflavin. When alkaline solution of riboflavin is exposed to UV rays, it converts into lumiflavin and gives greenish yellow fluorescence. When neutral or acidic solution of riboflavin exposed to UV rays, it converts to lumichrome. Next is niacin, vitamin B3. Niacin its study starts in 1913. In 1913, Funk isolated this from rice and yeast. In 1934, Ruffin and Smith discovered that crude extract of liver was effective against pellagra, and in 1937, Alpha-Gem isolated from liver as nicotinamide. Pellagra is a condition of niacin deficiency. Niacin or nicotinic acid is pyridine 3 carboxylic acid. It occurs naturally in body as amide, niacinamide, nicotinic amide. Amino group substituted into carboxylic acid to form amide group. The chemical structure of niacin forms nicotinic acid and nicotinic amide. Niacin is converted into active form of nicotinamide adenine dinucleotide, that means NAT, and nicotinamide adenine dinucleotide phosphate, means NADP. You should remember the full forms of both these, NAD and NADP. It's very important. Then what are the properties of niacin? Niacin is white crystalline solid. It is sparingly soluble in water, ethanol and glycerol. Niacin is heat stable. It can withstand 120 degrees for 20 minutes. Niacinamide is converted to niacin if heated with strong alkali or acid. Pantothenic acid is vitamin B5. It is essential to all forms of life. In 1939, it is isolated as its calcium salt from yeast by R.J. Williams. The structure of pantothenic acid, an acid consists of beta alanine, pentoic acid joined together by peptide bond. It is found in all living cells as coenzyme A, also called CoA. It is a vital coenzyme in numerous chemical reactions. What are the properties? Properties is it is pale yellow oily liquid that can only be crystallized as a sodium, potassium or calcium salt. Soluble in water, it is very much stable at 120 degrees for 30 minutes in neutral medium and is decomposed in acid or alkali medium. It forms ester with alcohol. Next is pyridoxine, vitamin B4. In 1938 it was isolated, in 1939 it was synthesized, and it was revealed that vitamin B6 activity in alcohol derivative pyridoxine and also its aldehyde pyridoxal and pyridoxamine. Pyridoxine contains pyridine nucleus, 2 alcoholic group and 1 phenolic hydroxyl group. By replacing CH2OH on the position number 4 of pyridoxine molecule with CH2, NH2 and CHO respectively. Two related compound pyridoxamine and pyridoxal can be formed. Three compounds are interchangeable. The active form of pyridoxine, pyridoxal phosphate. It is versatile decarboxylation and racemization. Properties: it forms white odorless crystal. The compound is readily soluble in water. When neutral or alkaline solution of pyridoxine is autoclaved at 120 degree for 30 minutes, partial destruction of vitamin occur. When the alkaline solution of pyridoxine is exposed to light, it is slowly destroyed, and pyridoxine produces a colored complex by reacting 2,6-dichloroquine chloramide. It forms salt with mineral acid and give violet colors. Cyanocobalamin and related compounds. In 1926, Murphy and Miner discovered that liver extract can cure anemia. In 1948, Smith and Parker and Rickes and co-worker isolated the crystalline vitamins. Dorothy Crowfoot Hodgkin determined the structure of cyanocobalamin. It is large and complex as compared to other vitamins, and the central portion is consisting of four reduced and substituted pyrrole ring surrounding a cobalt atom. This center is called Corin ring system. Below this system there is 5,6 dimethyl benzene imidazole riboside that is connected to one end to central cobalt and another end ribose moiety. The ribose moiety is connected to ring 4 of the tetrapyrrole nucleus through phosphate and aminopropanol. The cyanide is attached to cobalt atom, that is why the name is cyanocobalamin. If we remove cyanide, it forms cobalamin. What are the properties? It is crystalline, soluble in water. It is stable to heat at 100 degrees for long periods. It is an autoclave. Get converted into hydroxocobalamin. It is destroyed by heat at 120 degrees for 30 minutes in boiling. It is resisted in neutral and acidic medium but not in alkali. Aqueous medium of vitamin B12 get destructed in sunlight, and vitamin C when added it get destructed. Next is biotin. Egg yolk can prevent dermatitis. The egg yolk factor is called anti-egg white injury factor. In 1931, Shent Gyorby recognized it as vitamin and named as vitamin H. Melville isolated the vitamin from milk and named as biotin, and in 1943 it was synthesized. The structure is hexahydro 2-oxo 1-thiano 3,4-imidazole 4-valeric acid. Properties: it forms colorless needle-shaped crystal. Vitamin is sparingly soluble in cold water. This compound is sparingly soluble in alcohol. Biotin forms salt with alkali hydroxides such as NaOH. The compound is photostable and destroyed in acidic medium. Vitamin M. In 1947, Pfister and co-workers isolated from liver. This vitamin is called folacin or pteroyl glutamic acid. It consists of petroleum ring attached to p-aminobenzoic acid and conjugated with one molecule of glutamic acid. The molecule varies in structure by reduction of petroleum moiety to dihydrofolic acid called DHF and tetrahydrofolic acid THF. What are the properties? It is yellow spear-shaped crystalline substance. It is sparingly soluble in water. It is stable in acidic medium and destroyed by alkali. It can withstand the temperature of 120 degrees for 30 minutes at neutral pH, but if the pH is 1.0, folic acid loses 70 to 100% of its activity. When autoclaved at 120 degrees for 30 minutes, riboflavin accelerate the photooxidation of folic acid. Ascorbic acid. Holst and Froelich isolated this vitamin, and they reported that guinea pigs like man and monkey were also susceptible to scurvy. In 1917, isolation was carried by Zilva. In 1928, Szent-Györgyi isolated ascorbic acid from adrenal glands, oranges, cabbages, and he called it as hexuronic acid. In 1932, Glenn King isolated this vitamin from lemon juice and identified as Szent-Györgyi acid. It is named as vitamin C ascorbic acid. It exists as L-ascorbic acid in nature, and it is the derivative of carbohydrate and is a hexose sugar. It is most unstable and readily oxidized to DHA, means L-dehydroascorbic acid. What are the properties? It is white crystalline substance, freely soluble in water. It tastes sour. It is stable in air and light. In alkaline medium, it is oxidized very easily destroyed by cooking. This is very powerful reducing agent and can reduce Fehling solution. It is due to the presence of enol group at carbon number 2 and 3. It also reduces 2,6-dichlorophenol colorless leuco compound. It is used for chemical estimation of product, and both the reduced form means L-ascorbic acid and oxidized form L-dehydroascorbic acids are active. So I hope you enjoyed this video. I hope you will get the data and for your studies, and please study hard, and your examinations are near. And if there is any questions, any doubts, any suggestions, you can put it in comments. And if you want to ask any questions regarding biochemistry, nutrition, higher studies, registered dietetics courses, so you can ask that as well, or you can mail me at kavitakundra@gmail.com. Thank you so much. Stay safe, stay protected, and all the best for your examinations. Take care. Bye-bye.